Good morning, everyone. Welcome to our presentation of bacterial fruit blotch testing in the seed industry. My name is Sarah Curran. I'm the Marketing Communication Specialist for Eurofins Scientific. I'll be here to facilitate our presentation today. Before we get started, I have a few housekeeping items to take care of. This webinar is being recorded now. The recording will be available to everyone who registered, even if they did not attend. In 1 to 2 days, it will be emailed to you and it will be available on our website. There will be time for Q&A before the presentation ends. Questions will be answered via email if we do not have time to get to them. To submit a question to have our presenters address at the end of the presentation, you can use the question panel in your GoToWebinar dashboard. Here you can see you simply type the questions in, submit them, and we will select questions at the end of the presentation and I'll pitch them to our presenters. Speaking of presenters, today our presenters will be John Mizicko, Business Development Manager for Eurofins BioDiagnostics. A Colorado native, John Mizicko joined Eurofins STA Laboratories in 2004. He oversaw seed quality services, which was responsible for all seed health testing and germination testing. In 2010, Mizicko was named President of Eurofins STA and served in that position until 2015. When Eurofins STA merged with Eurofins BioDiagnostics in April 2016, his wealth of experience brought quite a bit to the merged operations. He's been involved in the California Seed Association, the Asia Pacific Seed Association, and the American Seed Trade Association, serving in various roles to help advance industry issues. Next, we will have Andrew Lyon, our Bacterial Fruit Blotch Supervisor for Eurofins BioDiagnostics. Mr. Lyon served as an Army Ranger for 3 years before earning his Bachelor's of Biological Sciences from Colorado State University with an emphasis on plant physiology and pathology. He worked in a CSU chemistry lab for 2 years before joining Eurofins BioDiagnostics bacteriology team. Today, Andrew oversees bacterial fruit blotch operations and testing as the BFB supervisor. Finally, we have Ravi Chitrampalam, research plant pathologist. He joined Eurofins BioDiagnostics in 2016. Ravi is responsible for developing advanced protocols for pathogens of importance to the seed industry. His expertise in plant pathology using both applied and molecular techniques, and he is currently working on molecular techniques that exclusively identify live targets of concern from seeds. Before joining Eurofins, Ravi served as a research scientist at North Dakota State University. He earned his bachelor's in agriculture in India, a master's in plant pathology from Punjab Agricultural University, and a doctorate in plant pathology from the University of Arizona. His research interests include biology, etiology, epidemiology, population genetics, and management of plant pathogens. At this time, I will hand the presentation over to John Mizicko, who will get us started.
Good morning, everyone, and thank you for joining our webinar on bacterial fruit blotch. We hope that it'll be educational for you, and we'll be anxious to see and respond to any questions that you have afterward. So let me get started talking about the significance of BFB to the seed industry. This organism was first observed in commercial fields in 1989 in the southeast, and virtually ever since then it's been found in almost every year, either in fields or greenhouses to some extent or the other. It has its greatest impact in the southeast of Georgia, Florida, but works its way up into the mid-Atlantic and northeastern states, as well as the Midwest into Indiana and Michigan. So there are outbreaks in those areas, especially in the southeast, almost every single year. It obviously can be very devastating to growers, everything from minimal losses up to 100%. Can be very devastating in the greenhouse, where greenhouse growers can lose an entire crop for a grower. And obviously, it has, over the past several years, created a number of lawsuits by growers in greenhouses, and these can run into the millions for the seed industry. It's gotten to the point now where growers and greenhouses really are expecting clean seed, and actually ask for certification that the seed that they are buying or putting into their greenhouses are clean. There's also been an impact to seed production practices ever since this has broken out. Part of it, as we found out where this pathogen lives and develops, in certain production areas, production companies have moved out of those areas. Places like China and India and Thailand, they've moved their productions from those areas that can create infected seed and moved into much drier climates, hopefully getting away from BFB. It's also gone to helping to certify stock seed lots. Growers that are doing third-party productions are asking for certification that the stock seeds that they're getting from the companies are clean. And obviously the companies themselves are testing stock seed to make sure they're sending clean seed into what they feel are clean areas. There's more routine inspections of fields where seed companies used to just inspect for hybridity and yield. Now they're inspecting for those plus any pathogen infections, and using things like immunostrips in the field to look at symptoms to determine if it's being caused by Acidovorax. And Andrew Lyon will talk a little bit more about immunostrips later on in the presentation. There's been changes in harvest requirements, where it used to be you'd go in and harvest all the fruit that was there because of the labor that went into producing hybrid seed. Now, most companies are only harvesting fruit that are absolutely clean or have no blemishes. And then there's also work being done in how to blend seed lots to make sure that a small infected lot is not being blended into larger lots. As you can see, it's really impacted the world. It's a global issue. Those countries indicate that both commercial productions and seed production areas, and my guess is that there are a few more that could be added to this map. So let's talk a little bit about the pathogen. It's a gram-negative biotropic bacteria, which means that it infects living material. It's motile with a single flagellum, and you can see a picture of a typical bacteria there. First isolated in '65 from accessions in the USDA plant introduction collection. My apologies for leaving out the D in the USDA. Really wasn't an impact until late in the 80s, where it really started showing up in fields. First originally named Pseudomonas pseudoalcaligenes, and then with showing that it infects, excuse me, cucurbits, they added on the citrulli to that name. Later was reclassified as an Acidovorax, and has recently been, the Avene name has been dropped out and just referred to as Acidovorax, citrulli, or AC. So a little bit about the seed transmission. It's obviously been definitively proven that it is. It's how it gets around. And if you remember the map of the world, that's really how it's gotten around to all those different locations, is through its seed-borne nature. Seed is a very efficient means of transmission. but it also gets spread by machinery and equipment and people and grafting. Warm, moist conditions are very conducive. That's why it's such a problem in, one, the southeast, and two, greenhouses. And it's been shown that even a very low level of infection in a seed lot can cause significant damage. Currently, there's no known eradication treatments. There are treatments that are used in the field to reduce down the infection level. There are treatments available in commercial fields to try to hold down the spread. There's also treatments that are used in seed production after the seed is harvested to try to knock down the amount of acidoborax that might be on the seed. So obviously, seed testing is a very important issue. It's extremely important for seed companies to test for AC in their cucurbit seed lots. All levels, we encourage all levels to be tested, not only commercial seed, but stock seed and foundation seed. We even have some companies testing breeder seed. It's been shown that phytosanitary field inspections, although they are good, they're not as good as they should be for detecting bacterial fruit blotch, simply because the symptoms in different areas are different, and oftentimes they don't look like bacterial fruit blotch. The industry initially said that a 10K test, 10,000 seed test, was good, but most companies have moved up to a 30K test, and that seems to be the most common, although we see companies asking for tests of 50 to 60,000 seeds. There are multiple methods available. Andrew will get into talking about that. You know, we basically have settled on the greenhouse grow out and the seed wash PCR. He'll go into more detail on those, but we've invested fairly heavily into both of those, and it's an area that we feel is the most significant. So this is really not the way to try to detect BFB in watermelons or melons. And Andrew will, as I said, will go into much more detail about the detection of BFB or acidoborax in seed lots. Again, the biggest problem is in melons and watermelons, but it has been found in squash and pumpkin and cucumbers. So I will at this time turn it over to Andrew and let him go into more detail about seed testing protocols.
Good morning, everyone. My name is Andrew Lyon. I'm here to go over the methods that we use here at EBDI and discuss a little bit about other methods that are used. I'm hoping that at the end of my section of this presentation, you'll feel confident in seed testing and being able to plant or sell clean seeds, so that this pathogen doesn't get you involved in a lawsuit, as John was discussing earlier. So there are four main methods used for the detection of BFB on cucurbit seed. We use the top two methods here, and I'll go into further detail on both of them in this presentation. The other two methods include seedling PCR, which is a relatively new method that we don't perform here in our lab, so I won't speak too heavily on it, and sweatbox PCR method that is not validated by NSHS. Here at Eurofins BioDiagnostics, we perform the seed wash PCR test and the grow out test. We recently earned our A2LA ISO 17025 accreditation for both of these tests. This means that you can be guaranteed traceability and consistency of testing methods and results. So we'll start it off with the seed wash PCR method. We use this method as a preliminary detection, and essentially, this is a new method that was just validated by ISHI and NSHS in July of 2017. We've incorporated it into our methods in September. Seed wash is an indirect method. It detects presence of acidoborax but gives us no information on the pathogen's virulence or viability. Because of this, all seed washes must be validated by a grow-out result at our location. Here's what a general seed wash looks like. Seeds are counted into subsamples of 2,500 seeds for large seed and 5,000 seeds for small seed, and placed into uniquely labeled bags. Seeds will be soaked in a wash buffer for 1 to 2 hours while being agitated by spinning. After soaking, a series of centrifugation steps will be used, and this will remove debris and pelletize remaining bacteria. That bacteria will be taken straight into DNA extraction. Here at EBDI, we use a QIAGEN kit for extraction. This results in quality DNA that's really free from contaminants and enzyme inhibitors. The goal is to have DNA that we can use directly in qPCR to get a quick turnaround and get a quick preliminary result. Here's one of our PCR stations. We use 3 primer sets for every PCR reaction, 2 specific for Acidiovorax and 1 used as a process control. This processing control is used on a known amount of XCV, and it shows us that the DNA extraction was successful. As with every ISO 17025 standard, we calibrate and proficiency test our machines once a year. Here's an example of a result from our PCR. These are just the results for the AC-specific primers. The XCVs, it's not really important in this slide. Both primer sets must display a positive result to be considered positive. Sample curves above display the positive sample from our greenhouse read, as well as a negative control curve. A negative control is used to verify that our reagents were not contaminated prior to use. This one, the positive from our greenhouse was so close to our positive control that they actually run the same curve, but as you can see, they separate here at the end. This is basically showing that we use a known Acidiovorax that's run through extraction as well, so that you can see that the result is the same. Now, in those results, you can see that the sample is positive and that the reaction worked, but what those results don't tell you is if the sample is viable and how virulent it is. That's why the greenhouse grow-out is imperative for confirmation and our preferred method of testing. We've been doing the grow-out test here at EBDI for about 24 years now. We chose this test because it's well established as the gold standard, and it's really the original testing method that all new methods are compared against. The grow-out test is accurate because it creates favorable conditions for the expression of the disease and tests a large representative lot of seed. As John mentioned, we use 10,000 to 30,000 seeds, but I've tested all the way up to 60,000 seeds. It just really depends on the lot that's being sent to us. Now, the results that we'll get from this test will address if the bacteria is viable, the level of infection, and virulence. All 3 of these will be reported to the client. This is how we prepare our greenhouses for the grow-out test. New flats and soil are used for every test. Nothing is ever reused. This is going to prevent the possibility of contamination and guarantee that your result is accurate. As you can see, we have a soil technician here loading soil into flats. We do this in a sterile environment, and we monitor this with 2 methods. We use an open-air media plate and a direct cotton swab plate. Essentially what this is doing is we're taking a plate of KB and we're leaving one open on the bench for a period of time, and we're directly swabbing the bench the soil is loaded into flats on. And then we check that after a few days of growth to make sure that no bacteria is growing. This ensures that the soil going into your flats is free from contaminants. To the right, you can see an example of our greenhouse map. We create this, and it's kind of like playing Tetris, where we load samples into the greenhouse. All samples are going to be divided by a large plastic barrier, and this keeps any contamination from sample to sample from occurring. Flats are then loaded into the house in accordance to the map, and labeled sticks are going to be placed on all sample borders. The seed lots are subdivided to allow for optimal growth. So you'll have 500 seeds per flat for melon and watermelon, or what we call small seed, and 250 seeds per flat for squash, as squash grow larger. After being loaded, we provide an 18-day undisturbed growing period. During this time, the technician that's watering the flats will visually inspect seedlings and update the manager with any abnormalities. We found things like positives, damping off, or low germination rates. We've seen positives pop up around day 7 and sometimes sooner, and we can address that to a client very early on in the grow. After the 18-day period, we'll evaluate all the seedlings. Technicians will look through each individual flat for symptomatic seedlings. This is going to be performed by physically leafing through every single flat and inspecting these seedlings. And it's very interesting because when a sample is positive, you'd be surprised at how far that infection will spread in a flat, and it really does catch your attention. But I will go into specific details on what we see in the greenhouse shortly. Each technician also carries picture reference cards during the read. This will give them a constant refresher, as well as positive controls being planted in the center of the greenhouse. And we all visually inspect those first to ensure that the test was valid, but again, to kind of refresh our eyes and get us trained. The sample processing kit to the right is brought into the greenhouse. This allows us to perform the immunostrip test on any lesions we find and get instant results. And I'm going to talk about the immunostrip test and how that works shortly. Now, the symptoms we're looking for are described as transdermal water-soaked lesions on the cotyledon leaves that will sometimes extend down into the hypocaudal. Lesions will typically have a yellow halo as the bacteria spreads. And this bacteria really does spread, as John was showing the picture earlier of the flagella. It's a pretty motile bacteria. After inspection, symptomatic tissue will be saved into additional bags to be used in the laboratory if necessary. So here's an example of our processing kit. To the left, I've included pictures of the positive control seedlings. You can see the lesions that I talked about. They really do move down the cotyledon of the leaf, and some of them extend down into the hypocaudal, and there's a better picture on squash where you can see that happening. And I've compared this to a sample that was negative for BFB and some of the symptoms that you might see in a greenhouse. Now, we normally wouldn't pull these. We know what we're looking for as far as symptomatic lesions, but I wanted to provide an example of some of the things that you might see in your fields. These are examples of seed pinch up here, where the actual seed coat has stuck to the seedling, and sometimes will cause lesions or a fungal infection. And there's a little bit of bleaching down here. Now, here's the immunostrip test that we've talked about. Our sample tissue was loaded into this tube and performed the immunostrip test. There's a control line towards the top and then a positive indicator line. You can see our positive control was successful in the immunostrip test. And these are performed on every new batch of immunostrips to make sure that they're working. Here's an example of watermelon seedlings. I have the sample tissue to the left and the positive control tissue to the right. On sample tissue, you'll notice another seed pinch example. This one was a little bit more drastic, and it does display the dark color, but the bacteria is not motile. We would occasionally will test seedlings like this just to confirm that there is no AC present. And here's some examples of some water spotting that will occur on the leaves, which is more common. And an untrained eye will look at this lesion here and think that it might be BFB, but as you turn the leaf over, you'll notice that it does not cross both sides of the tissue and it's not water soaked. In contrast to the positive control here, you can see that those lesions are traveling and water soaked on both sides of the leaf. Here's the examples of the immunostrip test down here, where the sample again displayed a negative result. And I took the tissue directly off of these seedlings to show this to you guys. And positive control again worked. Here's what we'll typically see on squash seedlings. Squash are a little bit more difficult to read as they can show a little bit more damping off, and they grow really quickly. So you'll have your true leaves growing out of the top and creating a canopy that kind of hide the seedling and the cotyledon leaves from view. So a little bit more difficult to read, but we've gotten quite good here at EBDI. These are some examples of symptoms that you might see on squash seedlings. This is a different bacterial infection. This is not Acidiovorax. And we have seed pinch symptoms here. And now if you look at our squash positive controls, and this image doesn't really capture it, I wish I could have zoomed this in a little bit better for you, but you can see the infection is spreading all the way down the hypocaudal. It was quite gross. And again, I've included pictures of the immunostrip test to confirm that our positive worked and our sample tissue was negative. Back it up one slide for you. Now, we plant positive controls in every greenhouse. We plant one control for every major species. This is going to include melon control, a watermelon control, and a squash control. The pictures that I showed you just previously were from each of these controls, and we plant them in bins like this to keep them separate from the rest of the greenhouse. Our positive controls are inoculated with A016. It's a proprietary strain that we created. A016 will grow on rifampicin, streptomycin media, while all other strains will die. What this allows us is us to plant positive controls in the greenhouse, but have a guarantee that that positive control species will never jump into other sample seeds. Every positive suspect client sample will have its symptomatic tissue macerated and spread on several plates of ripstrep. This ensures that cross-contamination does not occur. Notice down in the corner, we have a secondary greenhouse monitoring device. This is going to monitor relative humidity and temperature. Now, our greenhouses are controlled with a computerized system that monitors relative humidity and temperature, but we like to have a secondary set of calibrated devices to ensure that we're in compliance. When we have a positive suspect, symptomatic tissue is collected into 2 containers. Small pieces of tissue are placed into microtubes for immunostrip testing immediately, as seen earlier, and several additional leaves of that same symptomatic tissue are placed into a bag in case we need it in the laboratory. Now, our protocols from NSHS only require us to verify positives with 2 methods, with symptomology, immunostrip, and then the PCR to confirm. But here at EBDI, we use 5 methods to confirm our potential positives. We use symptomology, immunostrip, and PCR as required, but we also plate bacteria on selective media and perform pathogenicity testing from that media. After the immunostrip displays a positive result, we'll head into the lab, and we're going to plate that sample macerate onto several plates of selective media. The ripstrep that I discussed earlier, and you can see in this plate, there's no growth. There's a little bit of fungal growth at the top. But this will show that our positive control didn't jump and infect client seed. And we use BFBT as our selective media. BFBT is unique. As we add TTC, it'll form a red color as it's consumed by the bacteria. This is pretty specific for Acidiovorax. Some other species of bacteria will display this red, but I can run a positive immunostrip test and verify that Acidiovorax is on this. Now, what I'll end up doing is taking cultures from the selective media plate and perform pathogenicity testing. This test shows us the bacteria were isolated on selective media, and it'll show that it's infectious to a cucurbit host. In this specific pathogenicity testing, it was notable because we had several positive suspects from one client. And you can see the difference in these seedlings and how they were expressed during testing. Something important to plant pathology in general is cox postulus. This is how new infectious hosts are identified. And there are 4 main points. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. We do this with our grow out test. We satisfy this section of cox postulates because we do not find symptoms in healthy plants, but we do find disease tissue in unhealthy plants. Second stipulation is the microorganism must be isolated from a diseased organism and grown in pure culture. We do this with our selective media. The cultured microorganism should cause disease when introduced into a healthy organism. This is satisfied with our pathogenicity test. And the microorganism must be re-isolated from inoculated diseased experimental host and identified as being identical to the original specific causative agent. And we do this by isolating the bacteria back off of our pathogenicity plant and performing PCR and immunostrip testing. This is an example of what your final result will look like, positive or negative, or no evidence, as it's called. This certificate on the right is what will be used for import and export. Most countries in the world now require certificates like this for import and export of seed because of the virulence and danger that this organism causes. Here in Colorado, for our grow-out tests, we have 1,300 square feet of greenhouse space available. Each greenhouse can hold approximately 1,300 flats. In real terms, a 10K sample uses 18 flats, and a 30K melon sample would use 60 flats. We can process up to 2 greenhouses a week with a 2-day turnaround for cleaning, sterilizing, and reloading. As discussed, the greenhouses are temperature and humidity controlled with the state-of-the-art monitoring software and sensors that were updated in 2017. As we've seen increased need for testing squash and rootstock seed, we knew that we needed to provide more space, as squash samples use twice as many flats as other cucurbit samples. We decided to expand our program and add a 550 square foot greenhouse here in Colorado. And we also grew into California and added a greenhouse at our EBDI lab in California. This greenhouse is 4,000 square feet, which means it can hold up to 1,216 flats. This additional space is used to decrease our turnaround time and help us provide better service to our clients. Thank you, and I hope that you've learned a little bit about how we test the seeds here in Colorado and California. I'm going to hand off to Ravi, who's going to talk about molecular techniques.
Good morning, everyone. I'm Ravi, research plant pathologist. Next few minutes, I'm going to discuss the significance and challenges associated with molecular techniques and how the latest technology overcomes some of the limitations with these techniques. Molecular techniques such as PCR, qPCR, they have many advantages. They are very specific. They detect the target specific, they specifically detect the target in non-target backgrounds. So for this, you don't have to isolate and pure culture the pathogen from the seed. You can directly use the seed extract in qPCR. That seed extract may contain the non-targets and saprobes. So this qPCR specifically detects the target in impure seed extract background. And they are very sensitive. They can able to detect the level that cannot be detected by the traditional or cultural-based methods. So they can able to detect very few colony forming units or very few spores per ml of seed extract. So they are very good in detecting very low level of pathogens. And they are very fast. You can able to perform this test in a day or two. However, the culture, the traditional techniques, they may take several weeks to complete. So they are very fast, very good. Although the molecular techniques, they are very good, they have many advantages, still there are few advantages with these techniques. And they are too sensitive, depending on the purpose. It could be an advantage or disadvantage. For example, if you're dealing with the quarantine pathogens, so you want to restrict the introduction of the pathogen from one country to another country where there is no history of that pathogen. For that purpose, too sensitive is a really advantage. However, if the pathogen is already established, if it is commonly prevalent in that area, so in that sense, too sensitive maybe is a disadvantage, because for pathogens to cause successfully disease under field conditions, they have to be in certain number or certain level. We call it as a threshold level. These molecular techniques may able to find even the level below threshold. So that's considered sometimes a disadvantage. Another major disadvantage with the molecular technique is these techniques, they can tell you whether you have a pathogens or not, so positive or negative, but not necessarily they're going to tell you the biological activity of those pathogens. In other words, they don't tell you whether you have a viable pathogen or non-viable pathogen. That information is very critical. I can tell you, I can give you some scenario why that information is very critical. These days, seed industry, they usually treat the seeds to inactivate the pathogens before importing or selling to different countries. But if the importing countries use the molecular technique as a standalone or single method to test the pathogens, so that technique is going to show positive for the pathogen, even though those pathogens are inactivated by seed treatment. So that's a that's a false positive for viability. So based on that result, the importing countries may reject that seed lot. So that's going to cause severe loss to the seed companies. So that's a major disadvantage with the molecular techniques. Recently, scientists showed that indeed we can able to differentiate the live cells from dead cells used in qPCR itself using the DNA binding dyes such as ethidium monoxide and propidium monoxide. And the basic principle here is that in dead cells, the cell membranes are compromised, they are broken. However, in live cells, viable cells, the cell membranes are intact. So this dye specifically penetrates the membrane compromised dead cells and binds to the DNA covalently under the light conditions. And once it binds, that DNA is not going to be available for your qPCR. So how does it work? So you have the seed extract. Consider that seed extract contains both viable and non-viable cells, dead cells and live cells. So you're treating the seed extract with that dye before the DNA extractions, okay? So then the dye will specifically penetrate the dead cells and binds to the DNA. So now you're extracting the DNA after the treatment. So the DNA pool contains the DNA from both live as well as DNA from the dead cell. However, the DNA from the dead cell is preoccupied or intercalated with that dye. So that's not going to be available for qPCR. So whatever amplification you're getting in qPCR is specifically from the DNA from the live cell. So it's really, really amazing technology. And this technology has been successfully used in clinically important pathogen as well as plant pathogens. Recently, in 2016, Tyne et al., they tested the possibility of using this technology to differentiate the live from the dead acidovorax. And they conducted a series of extensive experiments in different combination of live and dead and different concentrations. And here the data from that study, and this study clearly showed that we can able to differentiate the live acidovorax from the dead acidovorax in qPCR itself. So that's a good news to the seed industry. So that's really, really a good technology and successfully used in acidovorax. As Andrew said, ISF recently released the seed extract PCR for the detection of acidovorax from cucurbits. And however, the seed extract protocol and the primer and probe used in this industry standard method is completely different from the one that used in the literatures. And furthermore, the PMA technology has not been tested with industrial standard methods. So we don't know we can able to differentiate the live acidovorax from the dead acidovorax using this technology. So we at the Eurofins BioDiagnostics, we initiated the preliminary study and see we can able to use these technologies to specifically detect the viable acidovorax. And here is our preliminary data, and x-axis the different treatments, and the y-axis the CT values from qPCR assay. And the CT values below 30 is considered as a positive for acidovorax. If it is above 30, it's considered as a negative for acidovorax. And you can see the treatments 1 and 2, there are live acidovorax. One is the live acidovorax without the PMA, the dye treatment, versus 2 is the live acidovorax with the dye treatment. You can see there is no significant differences between those 2 treatments, which means that the dye doesn't have any impact on the live cells. That's good. The treatments 3 and 4, those are with the dead cells. Treatment 3 is the dead cells without the dye. You can see that's the qPCR shows positive with the dead cell. However, number 4, the treatment number 4 is the dead cells plus dye. So when you added the dye, that treatment is you are getting the CT above 30, above the cutoff. It's clearly showing that that is negative for acidovorax. So this is our preliminary data, and it is clearly showing that there is a potential for using this dye under the with industry standard method to specifically detect the live acidovorax. So currently we initiated the extensive study to properly optimize this protocol to specifically detect the live acidovorax. Once it's optimized, this protocol will be shared with the NHS and the SHE to better serve the seed industry. Hopefully, it will be done in a year. That's all I have, and thank you for your attention. And we like to take your questions if you have any.
Yes, we would. Thank you very much, Ravi, and thank you, Andrew and John. Before we get started with the Q&A, oh, can you guys mute yourselves? There's an echo. Thanks. Before we get started with the Q&A, we have a few questions for you. So really quickly, we'd like to ask our audience, has your commercial production or seed production been impacted by bacterial fruit blotch testing before, or by bacterial fruit blotch before? All right, we'll give you a few more seconds to respond here. We'll close the poll and share our results. So a quarter of attendees say yes, their commercial production, seed production has been impacted by BFB before. So as a follow-up question, we'd like to ask, if you are one of the people who answered yes, what was the estimated economic impact of BFB on your operations, to your greatest estimation? Just so everyone is aware, these responses are completely anonymous. The information will not be shared, and we can't see who submitted which responses. All right, we'll give you a few more seconds to respond before we close. And about almost 70% fall in that 100,000 to 500,000 range. So that's a very significant impact to operations. So at this time, I'll pitch a few of our questions to our presenters. We have quite a few. Rebecca asked earlier in the presentation, how do you track the greenhouse conditions?
Conditions being relative humidity and temperature. Those are going to be tracked on 2 monitoring devices. One set is recorded on a computer system that physically controls the greenhouse. This is going to control everything from fans and cooling pads that will cool the greenhouse down in the summer, as well as heating units to warm the greenhouses up when they go below temperature. Relative humidity will be controlled with misting systems. Again, it's controlled by that same computer. If the relative humidity falls below 70%, it will start to turn on the mist systems. Positive controls, if that's what you meant, they verify that the greenhouse was in compliance with conditions. Again, with every single species planted, melon, watermelon, and squash, we inoculate those seeds and then plant them, and that'll show, if they show symptoms of acidovorax, and we'll immunostrip them and make sure that it is acidovorax on there. As long as they show symptoms, we conclude that the greenhouse was in compliance and the conditions were kept. Thank you.
Okay, and we have another question for Andrew. Can you share what primers you use in PCR for confirmation of greenhouse positives?
These primers are readily found on the NSHS website. If you want to send me an email, I can share precisely which ones we use, but they're going to be the same that you can see on the NSHS website.
Another attendee has asked, how did you choose your cutoff in qPCR detection at 30?
Yes, for that ones, we always use, before choosing the cutoff, we do several experiments with different concentration of a positive and non-target, and based on the values from the non-targets and also from the different concentration, you can choose the cutoff value. That's a standard procedure.
Okay. We have another question that's come in. One of our attendees asked, which of the several methods you mentioned is the most sensitive?
Well, I think if you're talking sensitivity and detecting colony forming units per cell, the PCR is going to be the more sensitive. But again, as Ravi pointed out, it's not telling you whether those cells are alive or dead. So, you know, we've always felt that the greenhouse corella not only is sensitive, maybe not to the level that a PCR is, but it is showing you that the pathogen is there and is active.
Do you want to talk about the potential for false positives in PCR too?
All right. Someone else has asked, how often are positive samples discovered?
So this is going to vary. It really kind of depends on what time of year it is and if clients are sending us seed that they know to be positive or that they have themselves detected positive in PCR. One of the problems with PCR, as Ravi discussed, is that it is too sensitive and it will occasionally give a false positive result. And so those clients will send those seeds to us and we'll grow them out and see if it is actually a true positive. But I would say in almost every read, we might find a positive or more lot of seeds. But I've seen up to, 4 or 5 or even up to 8 positive samples in one greenhouse read. It really just depends on where the seed's coming from and who's sending it to us.
Okay. I think we'll have time for just one more question here before we wrap up. Like I said before, questions that we didn't have time for will be answered via email after the presentation, and you will get a recording of this presentation emailed to you. So one last question before we go. Someone has asked, are there seed treatments available?
No, not really. Not that are completely effective. As I mentioned, in the production area, some companies will use a compound called Tsunami, and that is pretty good for reducing the infection of BFB. It'll knock it down, but there's not been any treatment found that is 100% effective in cleaning up seed. Good question.
All right. So at this time, we will conclude our presentation. I'd like to thank John, Andrew, and Ravi for sharing their time and expertise with everyone today. And thank you to everyone who took time out of their busy day to tune in to our presentation. Like I said, a recording of this webinar will be emailed to each one of you, and it will also be available on the Eurofins BioDiagnostics website within 48 hours. Thank you, everyone, and have a wonderful Thursday.
Thank you all very much.
Thank you. Thank you.